Good afternoon. I'd like to sincerely welcome you all to another webinar this year, sponsored by the Center for Biomodular Multiscale Systems and National Institute of Health, specifically National Institute of Biomedical Imaging and Bioengineering. We planned this webinar for about 50 minutes with, of course, time for questions at the end. So you will be able to either type those questions in the chat or ask them directly at the end. If you can raise your hand, then you will be identified to speak. Today, it is my delight to introduce Professor Steven Soper, our speaker. Professor Soper is a Foundation Distinguished Professor of Chemistry and Mechanical Engineering at the University of Kansas in Lawrence, Kansas. He holds an appointment at Ulsan National Institute of Science and Technology in Ulsan, South Korea, where he's a world-class university professor. He is also the director of the Center for Biomodular Multiscale Systems, short CBM². This is a P41 National Technology Center funded by NIH and NIBIP. The primary charge of the CBM² is to develop translational micro and nanofluidic tools for making health-related measurements. Some of these tools include integrated micro and nanofluidic system for sampling blood for presence of rare cells or extracellular vesicles, and the further molecular analysis of the cargo from those biomolecules. CBM² is tasked with development of microfluidic and nanofluidic devices made in thermoplastics. And the topic for today is very relevant to what CBM² is doing. Professor Soper will present microfluidics and nanofluidics for in vitro diagnostics and liquid biopsy analysis. The floor is yours, sir.
Thank you, Maggie. And hello, everyone. I didn't know if it's morning or afternoon, wherever you are. I'm happy you could join us. And I'm going to try to make this as very interesting as possible. And it's going to cover a lot of different areas. So hold on to your hats. We'll hear about engineering, chemistry, biology, clinical applications. And today I'm going to put this primarily in the context of in vitro diagnostics for cancer, but a lot of what I'm talking about will be very relevant for other diseases as well. That's for sure. So, in vitro diagnostics. I know what you're asking. What is in vitro diagnostics? Well, that's coming up. What I did want to start and point out is that precision medicine. What is that? And what kind of implications does it have on the technology that we're developing for managing a variety of diseases? So precision medicine, and a lot of you have heard about this, but basically what it indicates is that the molecular composition of a person's disease is unique to that particular person. That means that different strategies for treating that disease must be invoked by the clinician. And here's the challenge with that, is that you need the molecular signatures by which to make the appropriate clinical decisions, and you need the appropriate diagnostic test to identify those signatures and then assign the proper treatments once those signatures have been identified for a particular disease implicit in a particular patient. So this is quite interesting. If you look at this hit list right here, biologists, engineers, and then clinicians or pharmaceutical chemists, whatever you want to relate that to. And then down here, and the Precision Medicine Initiative was started right around 2011 by President Obama in the United States. And it has driven a lot of technology development, both on the therapeutic the disease signatures and the technology development. A really good example of that is next generation sequencing. That's been a technology platform that's been a really good driver for precision medicine. But look down here at all these different colored boxes. All of these have different meanings. And the first thing I just wanted to point out, this is a burgeoning field. In vitro diagnostic, the market is estimated to be about $150 billion by 2032. Even at this time, at 2024, it's still a relatively large market. And this is growing tremendously as we learn more about diseases and their molecular characteristics. All right. This is probably one of the most important slides that I'm going to show you today. And I want everyone, especially those that are well trained on the engineering side, to keep in mind, there is no such thing as a diagnostic test that can manage one's disease per se. There are specific tests earmarked for different stages of the management portfolio for any disease. And I've listed these here. The first one is screening. And the screening you can see here is large-scale testing. That means you're doing a lot of tests, and it provides only a red flag for deciding whether you need to go further on the testing for that particular patient, which then follows up with the traditional diagnostic test, which actually defines the organ of origin in the cancer regime, and then how this is done primarily through imaging. Prognostics, determining the fate of that patient. How do you treat that patient? What disease what therapy should be used for that particular patient's disease? And it was quite interesting. In the oncology area, these are primarily predicated on the use of a primary tumor source that's identified via imaging. And it can use liquid biopsy markers too. Response to therapy, and that's pretty self-explanatory. And then recurrence. You know, unfortunately, with many oncology diseases, you're not really cured per se, but you go into remission, and there's chances of recurrence. And this is what you want to be particularly sightful of. The other thing, and a really good case example of this, is colorectal cancer. There is a nice screening test for that. Using stool samples, Cologuard, you can see that little emblem here. Stool samples to look for proteins, blood, and even nucleic acids in the stool sample. And that just gives you a yes or no. If there's a positive result on that, a lot of times you'll go in and have a colonoscopy. In some cases, people don't even use the stool testing. They'll go right into a colonoscopy. And there's some challenges with that, with using colonoscopy as a screening tool. And we can talk about that after the talk if you want to have more questions on that. Then what about therapy? Well, in some cases, they're looking at certain mutations in the tumor DNA and to see if they can treat it with cetuximab. And that actually is a common therapy used for people with colorectal or colon cancer, colorectal cancer, or surgery as well. And then looking for response to therapy and recurrence by doing CT imaging to look at the tumor volume, primarily the primary tumor. All right. This is another important slide. Actually, all the slides are important, but this one, think about this. Cancer is a global concern, not just developed countries, but low to middle income countries. And let's just think for just a minute, what's the difference between, in fact, actually, this could be even in developed countries that have geographical issues. And a good example of that is Kansas. Western Kansas, it's all rural area. Eastern side of Kansas, it's mostly urban areas. Have access to high-end technologies to manage their particular cancer disease. So here's the challenge with this. To manage a cancer disease, you need access to high-end equipment as standard of care today. And many geographical areas do not have access to that. So a lot of people that have been diagnosed with a particular disease have to come, in the case of Kansas, have to come to Kansas City, Kansas, to be treated for their cancer at the major cancer center that's in Kansas City. And that becomes quite an issue because this could be for extended periods of time. So if you're looking for recurrence, that means you have to go back to Kansas City, Kansas, if you live out in Western Kansas, to look to have access to high-end imaging equipment like CT, MRI, and PET scanning. So that becomes quite problematic from just a logistical perspective. And response to therapy is done in real time during the therapeutic regimens, the chemotherapy. Well, the other thing too that you have to be aware of is that early screening, and let me just say a couple things about early screening. And I'm just going to say one thing. Early screening is just a single type of test that has in the portfolio of managing a disease that has different requirements associated with it. And let me just state a fact here. A screening test during the pandemic was primarily using these antigen antibody tests, this quick COVID test. Over a 2-year period of time from 2020 to 2022, there was 1.6 billion screening tests done in the US alone. All right, now you're getting a sense of scale. Screening tests can't cost thousands of dollars because it'll minimize the accessibility of that particular screening test and reduces the amount of population that even may be in a high risk group that would require screening. And that becomes formidable if the assay becomes complicated or it's costly. And it's very practical speaking. I didn't even talk at all about the technology and what has to be associated with a successful early detection or screening test, where we're either doing high-risk groups or population screening. And each one of these, based upon the biology of their particular disease, whether we're talking about colorectal cancer, breast cancer, ovarian cancer, has a different grade, which is on it, which is basically deciding the need for that particular screening test. In colorectal cancer, this is the easiest one because it's fairly well established, grade A, by this USPSTF agency. Breast cancer B, and ovarian cancer a grade D. And a lot of this is associated with the follow-up. What do you do with a patient once they've tested positive for screening? So that's a lot of how these are allocated. All right, so what goes on in the hospital? So if you're diagnosed with a particular cancer, you have a whole slew of tests done. And this is done in a clinical laboratory, and this is at UNC Chapel Hill. I went up there and took pictures and interacted with people at this clinical testing laboratory a lot when I was there. And again, you don't need to be a clinician to see the challenges with this. You have people, equipment occupying a large space, and a lot of these people are specialized or trained to do one type of test. So now you're seeing the complexity of this process. You have people doing specialized tests with specialized equipment that can be quite daunting. And doing this in a community hospital in rural areas is just not feasible. So microfluidics can have a great impact in eliminating these complex laboratories and relegating them to a more distributive form of testing, meaning you can do this in rural hospitals or doctors' offices. And microfluidics offers you a lot of capabilities that typically aren't associated with benchtop techniques. For example, point of care operation, new processing capabilities. And here's the easiest one, just to get back to what I said earlier from a screening perspective. Reduced cost, easily accessible, that means automation, and you can easily scale these up. But for those of you that are microfluidics people, be careful. Fabrication becomes an issue. That means if you're going to be doing 1.6 billion tests using your particular microfluidic, you better have an opportunity to scale that up to accommodate that a number of samples that need to be processed. Sampling statistics are so important. And the Theranos is a good litmus test to that fact. So, for example, if you're doing large-scale screening for early detection, what is implicitly true? I don't care what marker you're using. The amount of marker is going to be minimal. Much of it originating from the primary tumor is not going to show up in the body, for example, in blood, in circulation. So you better make sure you have enough sample to accommodate the sampling statistics. That is, you don't want to generate a false negative because you didn't have enough sample to look for those rare signatures of disease. And everything in the microfluidics domain is surface driven. High surface to volume ratio means the surfaces will play an integral role on the utility of that particular test, no matter what it is. But again, reduction in reagent sample consumption can be quite attractive because, again, you're making this more palatable for an insurance company to support or reimburse a particular patient on. So what about microfluidics? There's a variety of materials that you can use. We as a center primarily use plastics. Oh, for example, cyclic olefin copolymer, cyclic olefin polymer, polymethyl methacrylate, and so on and so forth. And here's the structures of these materials. We are not going to be talking about PDMS. And you're going to see a slide coming up why I am not going to talk about that today. And glass is another common material, quartz or fused silica, that's used for microfluidics. And there's some challenges with using either PDMS or glass from a manufacturing perspective and scale up. Plastics are very conducive to scale up. All right, so the first thing when you look at these plastics, the first thing you're going to say is, well, they're all structurally different. Absolutely. So now what you can do is match the material to the application need. And each application that you might have in a particular measurement or test has different specifications associated that may require a material that's more hydrophobic, more hydrophilic, has functional groups on it. And you can instill functional groups on a lot of these materials. And then looking at their thermal properties, their refractive indices, water absorption, transmissivity if you're doing optical detection, and so on and so forth. So why use polymers? And a lot of that I just outlined in the previous slide, but here's what I'm going to tell you about polymers, is that they have different properties, as I alluded to on the previous slide, and each one of these can be taken advantage of. But here's the bottom line on this. There's a production path for these materials. Clear production lines that can be used to generate a lot of devices at a very low cost with high reproducibility and high throughput. So, for example, injection molding is one way to do that. And injection molding is an established production line that was generated from the CD, DVD, and Blu-ray industry. Cost per chip can be low, potentially, about $1 per chip, and a high production per machine, about 1,000 chips per day. And there's other ways to generate these plastic chips as well, using, for example, hot embossing. Why is injection molding so important? And here's why. If you're looking at 1 billion tests that you want to carry out, and wouldn't that be great if our test showed up in clinical practice and we were selling 1 billion units per year? That would be awesome. But clearly, you want that cost to be low. And clearly, injection molding scales well with a high production requirements, whereas PDMS, elastomer casting, does not. It does not. So this is where you want to be careful with this scaling up. And when we get to a point where the need for that product is high, this is where this becomes particularly attractive. So this was a paper published by Holger Becker back in 2009, and I would encourage you to look at this. It's quite interesting paper. Is there commercial applications that are already on the market that are using microfluidics. And oh my gosh, yes, there is quite a bit, actually. This is a large market. Even in 2018, it had a $5 billion estimate and at a cumulative annual growth rate about 18 to 24%. This was predicted back in 2018, and it's following that particular trend. So this up to about 22 million by 2026. And a lot of this is being driven by the point of care market and the in vitro diagnostic area as well. And you can see materials, you can have here different components and applications. They're quite diverse. And it's not just the diagnostics, but drug delivery area, microreactors, and genomics and proteomics. This is basic discovery. So tremendous number of applications for this. And let me just, and here, this slide here just shows you the different companies that are doing microfluidics. And this is always amazing to people. Do you know NGS is using microfluidics? They have a flow cell. And actually what it consists of are these patterned wells that's made via lithography sitting on a microscope slide that has a cover plate that has fluidic operation. So you can input fluids in and out of this chip to do the complex chemistry that's associated with this. And this is called cluster amplification. And I'm not going to go through that today, but this pattern flow cell gives you these spots here that show up different colors that give you an indication of the nucleotide that's being added to this growing strand that's patterned onto this surface through the appropriate chemistry. So that's what you're looking at here. This pattern flow cell gives much better quality reads because of the non-random nature of those clusters that are generated. Each one of these little areas here is called a cluster. It's undergoing a bridge amplification step. Okay, there's another nanofluidic technology that's being generated. And this is this Oxford nanopore technology. And basically what this is a biological pore sitting on a silicon nitride membrane that's been fabricated using photolithography type techniques. And basically what you do is this nanopore here, you shuttle a piece of DNA through that. It gives current signatures that are indicative of the nucleotide composition of that DNA that's ratcheted through this pore at a rate that's determined by this polymerase enzyme that sits on top of this pore. And this is actually packaged up into a very small form factor instrument that has some quite unique aspects associated with it. And just kind of self-tooting our horn, the center is also developing a nanofluidic, a chip-based technology for single molecule sequencing as well. And you can see that right here. There's a number of things that are going on here. One is an enzymatic step that actually takes the DNA, chops it up into its monomer units, and then uses nanoelectrophoresis and resistive pulse sensing to look at the composition of those DNA fragments by clipping it using a reactor that consists of an exonuclease. And I know I said a lot there, but this is made by fabrication techniques, in particular photolithography, ion beam milling, and then these devices, once they're fabricated, have dimensions from several tens of nanometers to several tens of micron, are injection molded to accommodate the mass production requirements for in vitro diagnostic applications. And this is a paper, this came out of Dr. Sunguk Park's lab at Louisiana State University. And this is a paper that kind of talks about how that technology works. And that's about all I'm going to say about that. If you're interested in learning more about that, we have people on part of the center that can help facilitate that discussion with you. But let's go back into liquid biopsies. What are liquid biopsies? And basically what that means is that you're sampling the liquid part of the body, and that could be serum or plasma, the whole blood, the saliva, urine, or even tears. But why do you want to use liquid biopsies? Well, because they're minimally invasive to non-invasive, gives you the opportunity for frequent sampling. And the other thing that's so important, at least in the oncology perspective, they're not just looking at the primary tumor, but metastatic sites as well. So you're getting a more holistic overview of the tumor and the cancer, that's for sure. And because of the minimally to non-invasive nature, they're more accessible markers that can be used for frequent testing that sometimes may not be advisable if you're using solid tissue biopsies. So that means that the technology can be more disseminated into the general community by using liquid biopsies, which makes it particularly attractive for what I call distributed medicine, getting tests out of the cancer centers, the high-end cancer centers, and into more remote geographical areas. And these liquid biopsies they consist of several different markers: circulating tumor cells, cell-free tumor DNA, or extracellular vesicles, but they can be used for a variety of different types of management steps in the entire management portfolio. Those are the tests that I showed earlier in my slides. And this was published back maybe about 9 years ago, but circulating tumor cells, they have very high clinical figures of merit in terms of the positive predictive value and their negative predictive value compared to things like PSA, CA-125 for ovarian and prostate cancer, respectively, and anything else that you can show up. In fact, actually, we published some papers on using circulating tumor cells as a prognostic marker or response to therapy marker instead of CA-19-9 for breast cancer. So that's actually quite interesting as well. Oh, yeah, so that's the next thing I just want to remind everyone. When you're developing a test, make sure you understand what domain that test fits under. That means, is it a screening test? Is it a diagnostic test? Is it a prognostic test? Response to therapy, or recurrence? Each one of those has different clinical and analytical figures of merit that require different components to be engineered into that test to accomplish those figures of merit. Please keep that in mind. They're different. No matter what reviewers may tell you, they're different. All right, well, what about CTCs, circulating tumor cells? And there's a variety of ways to isolate those. So that's the other thing I need to back up and remind you. When you have, let's just take blood as an example. In 1 ml of blood, there's 10 to the 10th red blood cells, 10 to the 6th or 7th white blood cells, and maybe 1 to 100 or 1 to 1,000 tumor cells. So what does that mean? I don't care what liquid biopsy marker you're using. You typically have to use an enrichment approach to make the downstream processing palatable, required. So there's different ways to isolate circulating tumor cells, either using the biology, that is using an affinity-based approach, or physical properties. For example, the size, the density, and the electrical charges associated with that particular cell, and how they differ from cells, blood cells. And so you can think about this: size. Well, what could you do to separate tumor cells from blood cells, whether it be red or white blood cells? If you use size, you could use a simple little filtering-based approach, which is, from an engineering perspective, pretty easy to do. But those tumor cells must be larger than the blood cells. Otherwise, this becomes intractable. And the place that this breaks down is for the leukemias, especially in T-cell-based leukemias, because the T cells are comparable in size to the white blood cells, and filtering becomes impossible with that particular format. But you can enrich with those cells using techniques, for example, affinity-based approaches, where you're using an antibody to capture those tumor cells that have a unique molecular composition that's not found in the blood cells. So one common technology to do that is called the cell search approach, which was developed, and one of the colleagues in our center, Andy Godwin, actually was part of the initial effort to bring this to market. And basically, it's a magnetic bead-based approach. You use ferromagnetic beads coated with an antibody. You mix those beads with a blood sample, and then magnetically pull the beads down because of their magnetic properties. Once the cells are express an antigen are incubated with that blood sample. And you can see, well, a lot of work was done for the breast cancer. This was published way back in 2004. So a little bit of this is outdated, but you're just getting an idea of the number of CTCs that show up, and they're much dependent upon the disease at which you're targeting. Now, let me say something about that. I said it's dependent upon the disease. Not always. It's always dependent upon the technology, the analytical approaches that you're using to isolate those particular CTCs. So the answer to this can be very, very complicated. And then once those cells have been isolated, you can do what's called immunophenotyping to see which ones you have. This tumor cells should be nucleated. They express cytokeratins, and they do not express CD45, which is an antigen that's always shows up for the most part in white blood cells. So when I started talking about this work about 15 years ago, the common thing I heard was, well, Steve, there's not many CTCs in blood. And I found this paper was published in 1975, and this disputed exactly what people said. Cancer patients shed 3.6 times 10 to the 6 tumor cells per day per gram of tumor throughout the tumorigenic process. That's a lot. That is a lot. So they are very abundant. In many cases, the challenge is their decomposition when launched into circulation, the changes that they undergo when they're in circulation, meaning they express different antigens that could be used to isolate them, and the clearance from the body, or the analytical technique that's used to isolate them, or all of the above. And we use an antibody. In fact, actually, one of these is looking at an epithelial cell adhesion molecule that's attached to the surface of microchannels that carries an antibody onto that surface that's targeting this antigen found specifically in the tumor cells, but not primarily in the blood cells. And in most cases, this is for the solid tumors only. Let me just point that out. And there's very few, if any, EpCAM-bearing cells in circulation. All right, so what do we know about microfluidics? One of the things we know about microfluidics is they typically have low chaotic flow. They have laminar flow. Laminar flow, because the Reynolds numbers are so small. Who cares? What does that mean? Well, if we put antibodies that are looking for tumor cells based upon the antigens they express, and we launch it through a microchannel, the antibodies are poised on the surface of this microchannel. You can see that because of the laminar flow and the low Reynolds number, some of these cells never reach the wall. That means the recovery is going to be low. But if I just do a simple little thing and putting on this herringbone structure into this, we generate what's called chaotic flow. And you can see we've disrupted the flow lines of these cells as they're moving from right to left in this microchannel. And now we give them a better opportunity to interact with those antibodies poised on the surface. That means the recovery is going to be higher. And this was generated by George Whitesides and his group. Oh gosh, this is Shannon Stott who published this paper, but George Whitesides presented these are called herringbone mixers, maybe about 20 years ago or so. And this works very nicely. that we see from this, just pointing out some things here, the capture efficiency, and if you're looking for only a few cells, you better have a high capture efficiency, is highly dependent upon the flow rate, and the channel height, and other analytical figures of merit. And for example, the expression level of the antigen on the cell. If the cell doesn't express the target antigen as dictated by the antibody that you're using, you're never going to catch it. And the reason I bring that up is because a lot of these tumor cells, when they're released from the primary tumor into circulation, they undergo phenotypic changes. So some of the antigens that you think should be there are actually not there because of these transitions that these cells can undergo. And that just hits home on what engineers need to do when they're designing devices for in vitro diagnostics. Consult with your biologists and your clinicians. First of all, clinicians, make sure the problem you're working on is important clinically. And the biologists, make sure you understand the changes the markers can undergo during their livelihood. So one of the things that we have done is actually developed an affinity-based approach for isolating circulating tumor cells, and it's based upon a fluidic network. And I'm going to talk a little bit about this just to give you an idea how these are designed. But you can see these blood cells and the tumor cells moving hydrodynamically through these channels, and they're getting snagged up by the antigens that are binding to these antibodies poised on the surface of this device. So this is what this device looks like. It consists of these sinusoidal channels, a whole bunch of them, over about 150 of them, with a common input and a common output to make it easily accessible in terms of the fluidics. So we're launching whole blood through these channels that are roughly about 25 microns wide and 150 microns deep, with the antibodies poised on the wall. All right, so whenever you start a test for a clinical application, the first thing you're going to hear from reviewers is, what about non-specific adsorption? And that's ubiquitous across the engineering domain. That's the first question that spills out of people's mouth, especially for microfluidics, because the surface to volume ratio is so high. So look at this slide right here. We've purposely engineered the surface after blood is transported through this device to minimize any non-specific adsorption artifacts, to mitigate that issue. And I always show this when we submit proposals, so I don't get a review back and it says, well, you didn't talk at all about non-specific adsorption. Well, yes, we did. And here it is. So now let's just go quickly through this. We're launching whole blood through that. What happens when blood is launched through a very narrow channel? Well, the back pressure is going to be high because the blood has some viscosity associated with it, depending upon what's called the hematocrit level, the amount of blood cells that are in this. We want to be able to process a large amount of blood in a short period of time, so these channels are narrow to increase the interaction time of the solution-borne cell with these surface immobilized antibodies. And keeping in mind that there is laminar flow in this particular device because the Reynolds numbers are so low. We've created also many channels here to increase the processing time. And why is this sinusoidal nature necessary here? Well, when you take a fluid loaded with particles, cells, and you launch it through this type of device, there's centrifugal forces that are generated that'll push the cells up against this channel wall on this side of this curve. You can see right here where my pointer is. And that pushes these cells against the wall, and they'll roll along the wall, giving them opportunities for interacting with these surface-bound antibodies. So the purity the recovery is high. We can process about 1 ml of blood in roughly about 15 to 20 minutes with high recovery. And then the purity is also important as well. And this is just an example of that Dr. Vitek published a paper in Nature Precision Oncology showing how this technology works for isolating tumor cells from a variety of diseases: epithelial ovarian cancer, breast cancer, colorectal cancer, pancreatic cancer, prostate cancer, and healthy and non-cancer patients, and then doing the appropriate immunophenotyping to show that, yes, indeed, we are isolating cancer cells, and then putting in an additional marker, not just this epithelial one, but this mesenchymal one that's indicative of those cells that are undergoing biological changes after they've been released from the primary tumor site. And there's a whole slew of markers that you can use to isolate CTCs, and many of those are shown on this slide. And what this slide is just showing you, this filter-based approach, which is, again, a microfluidic that has a very narrow distribution on pore sizes that allows the white blood cells, the white blood cells in particular, red blood cells are typically removed before you put them through this device. And it captures the tumor cells because they're much larger than the white blood cells. And this can be made in PDMS with an 8 micron pore size that's made via casting against a relief, for example. All right, what about another liquid biopsy marker? And these are the extracellular vesicles. And these are becoming very popular. And I'm not going to give you an exposition on extracellular vesicles, except just to point out that these are roughly, again, depending upon the type of exosome that you're talking about or type of extracellular vesicle you're talking about, they can be very small as 30 microns up to be up 30 nanometers, 30 nanometers, up to about a micron in size. And there's several different types of EVs. There's the exosomes, which a lot of you are familiar with, the microvesicles, which have a different biogenic source, budding off the membrane, then the exosomes, and the apoptotic bodies. So each one of these is different and carries different information. These EVs also carry molecular information like RNAs, carbohydrates, proteins, and they have a lipid bilayer shell. So you can, the challenge with the EVs compared to the cells, it's not such a hard one to realize. It's that they're smaller. So enumerating them becomes much more problematic because of their intrinsically small size. So one way to do that is using transmission electron microscopy. And that's what you're looking at here, EVs that are imaged using microscopy, transmission electron microscopy. And then to enumerate them, one way to do that is to use nanoparticle tracking analysis. And basically what you're doing is you're shining a light on a microscope slide that has the EVs in them, and they scatter light. And then tracking these particles will give you an idea of their diffusion coefficient, which you can relate back to their size. So that's what this does, the size distribution and enumeration particle size numbers by looking at the amount of light entities that you're seeing in this particular slide. And then looking at the protein content by using Western blotting. And that's here. Here's a EVs from HeLa, and they're generating this heat shock protein 70. A lot of the extracellular vesicles, in particular the exosomes, will generate what's called these tetraspanins. For example, CD9, CD63, and CD81. So if you're particularly interested in looking at exosomes, then you'll look for those tetraspanins, CD9, 63, and 81, using a technique called Western blotting, and that's shown here. And a lot of times there's this agency or this association called the International Society for Extracellular Vesicles, and they want to follow a path to verify that indeed you're analyzing extracellular vesicles and not some other type of nanoparticle found in a biological solution. Okay, I'm going to skip through that pretty quickly. So what I showed you is microfluidics can be used to isolate circulating tumor cells. What about the nanoparticles? What can you do there? Not so easy. And that is because the physics of their transport in fluids, especially in a microscale environment, is different than the cells, based solely on the size of the particles, their density, and their electrical properties. So one way to isolate EVs is not using the cell search technique, but using ultracentrifugation. And here, we've done this, and here's the challenge with this. In fact, this is a machine in the center's lab, is it's a long and arduous task, 6 to 12 hours to isolate the EVs with 5 to 20, 40% recovery if you're lucky, with not so good purity. 5 to 25% recovery, you can see, and it isolates all the EVs. All right, I must stop here and say one very important thing. EVs release continuously or secrete continuously from cells, EVs, diseased cells and non-diseased cells. And the challenge is, especially for early stages of disease, the disease cells, which carry the relevant biomarkers that you're interested in, are much less abundant than the EVs coming from non-disease cells. So this technique here will get all the EVs, irrespective of whether they're diseased or not, in terms of their source, cellular source. So that becomes very problematic when the diseased EVs are a vast minority in a mixed population. So we've developed a device to do that. It's not showing this sinusoidal architecture that I showed for the CTCs, but it's a vast array of pillars that have antibodies attached to their surface. So we can easily attach the antibodies onto the surface of these plastics. And we have a webinar that we've given on surface modification of plastics that I didn't share with you, but you can freely go to our website and go back and look at that webinar on surface modification of plastics to get an idea of how that's done. But we can easily put onto the surface antibodies with the appropriate spacing, the size, and the density of these pillars to give us good analytical figures of merit. And what is that? High recovery, large dynamic range, and high purity. And you can get an idea what's going on here. Let's see if I have this. Oh yeah. Is as the flow is moving through this bed of pillars, we require the extracellular vesicles to interact closely with these antibodies before they're snagged up. And that does create a little bit of a conundrum in terms of the isolation. That's kind of manifested here. So I'm just going to show you this slide here. So any affinity-based isolation, that's why I'm showing you this. It's not just true for our device, but any affinity-based isolation approach where the antibodies are attached onto a surface. If you're using particles, nanoparticles, or large cells, they have to be oriented properly in order for that antibody to interact with that antigen. And if you come over here, you can see, okay, this particle interacted with the surface, but it didn't induce a capture. The X marks the spot where this capture is appearing. So it wasn't oriented properly with the antigens that were found on that particle. Surface interactions here, here, and here, until finally it generated an association event. And that's what's actually going on here. This is true for any type of solid phase enrichment process. So whenever you start with EVs, the other thing that you're interested in, so now we have a microfluidic to isolate them and look specifically at disease-associated EVs by targeting, just the same way we did with the cells, specific antigens. So the first thing you do is look at the cells and look at the EVs to see if they track each other in terms of their molecular content. And we have two different cell lines down here, an epithelial and a mesenchymal cell line, and look at expression differences for different genes to see if they're tracking each other in the cells, with that in the EVs. And they do for the most part, but there is subtle differences, that's for sure. And the biology of packaging RNAs into EVs is quite complicated. And in many cases, differences in the EVs, RNAs can be quite different than the cells from which they were sourced. And that puts severe challenges on the downstream processing. And then looking at the isolation of these, looking at the ultracentrifugation, how these look compared to the microfluidic component, you're going to get a pure fraction of the diseased EVs, and looking at the size distribution by using nanoparticle tracking analysis and TEM analysis. And this is coming. This is one of our colleagues, Andrew Godwin. This is a device that was generated for using microfluidics to isolate EVs, and it's using a bead-based technique. So the reason I brought this up is because a lot of things you can do on the bench, you can also do in a microfluidic. So I'm incubating the EVs with beads, microscale beads that are ferromagnetic, and they're coated with an antibody. So I can incubate the plasma or the blood, particularly plasma, with these beads, pulling them down magnetically. And then I can do some downstream processing on these. For example, looking at their protein content by adding the appropriate reporters into this cocktail carrying those exosomes. And clearly you can see relative abundance of EpCAM producing EVs for non-small cell lung cancer, ovarian cancer, and then compare that to the ultracentrifugation of non-small cell lung cancer. You can clearly see differences in the size distribution of these EVs, and then aggregate size fraction, volume flow rate, how that affects the recovery, and then using a microscopy technique to look at certain antigens that have been incubated with antibodies carrying a fluorescent reporter, an antibody for these particular markers up here that I'm showing. Yeah, and this can actually be then put to use for doing clinical studies. And again, let me warn you, clinical studies, you must be able to do the biostatistics and the power analysis before you ever invoke any clinical study to make sure the number of samples that you use are justified, and they're going to answer with high precision and clinical figures of merit the particular question that you're trying to delineate. And you can see just looking at this, and I'm running out of time here, so let me just say this is looking at non-small cell lung cancer in healthy individuals. You can clearly see that the vesicle abundance is increasing for these non-small cell lung cancer patients versus healthy individuals. And that's clearly shown in this box plot down here as well. And then looking for markers, those that may have more of a cancer indication like EpCAM, and then these tetraspanin markers here as well. And then the final thing I'll say is, this is the challenge. Whenever you're doing a molecular assay, it's never a one-step process. And I was just at a meeting last week, and there's only very few examples where you have what's called a dipstick test. That means that you dip the sensor into a clinical sample and it gives you the result. And the only one that I know of is a continuous glucose monitor. People can wear the patch and get a continuous monitoring of their glucose, the glucose level. But what do we know about the glucose level in blood? It's pretty high. It's micromolar concentrations. So it's fairly easy and straightforward to measure. But all the things that I showed you today in terms of doing the molecular processing require multiple steps in the diagnostic or the assay, in vitro assay. For example, selection of the particular cells, counting those cells, and then doing a molecular analysis on the content of those cells, or EVs for that matter. So we're building these modular systems that allow you to carry out multi-step assays in a fully automated fashion and do that with high reproducibility, even for people who are not well trained in the art of doing microfluidics. Keep in mind, the people that are going to be doing these measurements are not your graduate students. They don't know how these things work. They spend their livelihood on the bench work and optimizing the performance of these devices. These are done by clinicians and nurses and technicians that are not well versed. So automation and simplicity in the operation are paramount. I'm going to skip this. I'm going to skip this too. And how we use that, those multitask integrated systems for doing clinical studies, looking at response to therapy in PDAC patients. All right, so that's what I was going to share with you today, and I'm a little bit over, not so bad. And I would encourage you, if you have further questions on this technology, is to go to our website, which is listed right here, and our Facebook page. The other thing, as a center, our mission is not just to do research, but disseminate the technologies that we develop. So all the plastics manufacturing, and you're seeing a lot of that activities going on here in our clean room on the KU Lawrence campus, and the injection molding machine, we can help facilitate you learning how to transition your potential assays from using a PDMS-based device to a plastic. And I'm going to warn you, a simple translation from PDMS to a plastic is not straightforward. And the major driver of that statement is the fact that the surface chemistry is completely different, which may require a different type of strategy to do the particular device application. So just keep that in mind. And we have collaborators at a variety of universities that are shown here, and they're actually willing and offering up help. We also have exchange programs where you can send students to the lab to learn some of the things that we do here, and we're happy to do that. We don't keep everything in-house. Our job, as mandated by the NIH, is to disseminate our technology into the community, and that's what we strive to do. So why don't I stop there and answer any questions that may show up in either the chat box or you can raise your hand. So I will stop. Thanks for your attention and be happy to answer any questions that you might have.
Thank you, Dr. Soper. Yes, we have time for a few questions.
Any question will do. There's no such thing as a stupid question.
Dr. Soper,
yes.
can you hear me?
Yes, I can hear you.
Curious. Okay, I was curious about the C2C number that you mentioned, because you mentioned the study that you had. You mentioned a number of 3.6 times 10 to the 6 cells shedding from the tumor, but we also see all those references about having just 1 to 10 cells per mil of blood. So I was a little bit confused about that. I remember you mentioned also some changes that those cells could undergo, including the absence of the antigen. So I was wondering if this is the thing, they are present but not being able to identify it. I'm not sure if I understand correct, so I would like to hear a little bit more about this.
Okay, all right. And I have forgotten. Who can tell me? So the numbers that Shayla mentioned were per mil. How much blood is in a human? Is it 5 liters?
Yes.
Is that right?
Yes.
5 liters of blood. Okay. I said 3.6 times 10 to the 6 tumor cells. Was it per mil of blood?
Is it per gram of a tumor within 24 hours?
Yeah. I never said per mil. So the first thing you have to remember is that those tumor cells are being disseminated into that 6 microliter or 6 liters of blood. So that's the first thing. They're being diluted because of the total volume of blood. They're being continuously removed by kidneys or shear forces on the cells. And then the analytical technology that's used to isolate them. You're never going to reach that 3.6 times 10 to the 6, number because of the simple fact that you're only pulling out a mil of blood per patient, analyzing a mil of blood, not the full 5 to 6 liters of blood. So that's the
Thank
first
you.
thing. And again, remember, the analytical technology, no matter what you're doing, is never going to be 100% efficient. So you're going to miss some. And the biology accounts for some of the loss of critical material as well.
Makes sense. Thank you, Dr. Soper.
Anything else?
Yeah, I'm looking, and if I'm missing you and you're raising a hand, please go ahead and speak.
Hi, this is Lynn Pulliam.
Oh,
Oh, hi,
hello.
Lynn.
Hi. Sorry I was late. I got the wrong Zoom. Obviously, I'm interested in exosomes, and I'm wondering, is there any way even that you could do now or maybe in the future where you isolate a extracellular marker like a CD9 or tetraspanin, and then somehow enzymatically test for an internal protein? Would that be able to be done on one chip, or would you have to go, isolate your exosome by a tetraspanin first, and then maybe put on a second chip and somehow lyse the cells, lyse the exosomes?
And you know what, Lynn, the point that I was trying to make in this slide right here was that chips are not relegated to doing only a single step.
Okay.
They can do multiple steps. For example, this chip here that came out of Andy Godwin's lab and his colleague Yong Zhang was doing protein analysis. So they capture the EVs on a magnetic bead, and then they incubate it with antibodies that are carrying a reporter targeting specific antigens on the EV. And then and that's done here, protein capture, magnetic beads, and so forth, and then image those using a fluorescence microscope, for example. So like in your case, Lynn, there's no reason that these O-link assays cannot be done on a chip as well. We just never got there. And to automate that process. And here's the advantage too, Lynn, is that you scale it down. When you scale it down on a microfluidic where you're using much less reagent, the cost of that assay drops precipitously, as you might suspect. The other thing that we're doing right now is looking at the intravesicular contents. We're planning to do this, to look at the RNA content using a CRISPR, the microRNAs in particular, using a CRISPR-Cas13 readout and gene editing system to look at microRNAs and develop simple readout strategies for looking at those particular products that are generated from that. So anything you can do on the bench, it can be transitioned for the most part onto a microfluidic. And these things can be generated to do multiple processing steps in series.
Okay, so you could, for example, lyse an exosome once you've isolated it with a tetraspanin or whatever, and you could actually query for either proteins or miRNA or almost anything. Yeah,
absolutely.
I think that's by far the best way to do that.
Yeah, absolutely. I couldn't agree with you more. It's not only scaling the total volume down, but scaling the number of assays that you do by putting multiple channels on a single chip. And this is very simple to do, very straightforward to do. And this is a good example of that.
Okay, I have another question. Because I am interested in the CRISPR and I'm on a advisory committee for that, why couldn't you use the microfluidics to query whether or not there's any virus left? Because as you know, once we do the CRISPR or whatever, is there any virus left that's coming from the reservoir? This is a big deal right now. It's how can you test? And I would think that microfluidics might be a better way because again, you might have the proteins circulating, but there'll be very few. I'm just trying to figure out if microfluidics could be helpful after a CRISPR to determine if you've gotten rid of all the HIV. I mean, like a tumor cell.
Yeah. Oh, sure. You can. There's no reason you can't do that. If they're very low abundant, then enrichment might be necessary in order to build up the resultant signal, but that can be done using a variety of microfluidic techniques as well. So enrichment was done here through this immunomagnetic approach before they ever tried to do the readout using fluorescence of these particular protein markers.
Okay.
So this just requires consultation between the clinician or the biologist and the engineers to make sure we're covering all the necessary aspects of the assay that's required and looking at all the biological nuances that may be adherent in a particular assay. The other thing that's really, really important too, and I talked to Maggie about this morning, is always think about this. What are the figures of merit that you require, both on the analytical and the clinical side? So one of the things that are quite pervasive in the engineering side is, oh, this marker, I have a new world's record limit of detection of 1 femtomolar. So the first question I ask them is, okay, what's the actionable level, the clinical actionable level of that marker in solution in a body fluid? Oh, it's from 1 micromolar down to 1 nanomolar. Well, then why are you wanting to detect 1 femtomolar? It doesn't make sense. There's no clinical need for that. What is required if you're looking at expression levels is the change, the analytical sensitivity or the change in concentration of that. So there you want high analytical sensitivity, which is related to the slope of the calibration curve. So consultation with the clinician and the biologist and the engineer are important. So you got your ducks in a row in terms of the figures of merit. All of that will drive the engineering of the particular device. So the exosome thing that we have built has a really high surface area. It's using a lot of antibody, but it has a high surface area, gives us a huge dynamic range if we want to scan for changes in expression of a particular RNA or a particular protein, which demands a high a large dynamic range with high analytical sensitivity.
Okay, great. Thank you.
Thank you so much. I don't see any requests for questions. So thank you so much, Dr. Soper, for presenting today. Thank you all for joining us and listening. I'd like you to stay tuned. We are planning for 2 additional webinars in December, so stay tuned for the advertisements we're gonna send. I wanna wish you the pleasant either morning, afternoon, or whatever the time zone you're residing, and see you soon. Thank you.
Thanks, everyone.